Hi everyone, my name is Amanda Del Rosa, and today I'm going to be presenting my thesis to you titled Raising the Wall, the Effects of Artificial Flood Infrastructure on Soil Properties and Greenhouse Gas Fluxes. So first I'm going to start off with a little bit of background on salt marshes. So they are highly productive environments that supply a number of ecosystem services, and some of these include water quality regulation, nutrient cycling, nursery grounds, coastline stabilization, and they're also one of the most significant global carbon sinks due to their extensive carbon sequestration and storage capacity. And so here we can see salt marsh importance to the global carbon cycle. This figure displays mean long-term annual rates of carbon sequestration in both terrestrial forest and coastal ecosystems. And so on the whole, we can see that salt marshes contribute substantially to long-term carbon sequestration. And now to detail the mechanism a bit about behind their carbon capture. So healthy natural salt marshes have a high rate of CO2 uptake during photosynthesis. And since salt marsh soils are often water saturated, this anaerobic environment causes slow rates of organic matter decomposition, resulting in significant carbon storage. So since they are frequently inundated with seawater through tidal exchange, there are high concentrations of the sulfate ion. And so this ion is more energetically favored by microbes, so sulfate reduction tends to outcompete methanogenesis as a metabolic pathway, resulting in low methane production and thus low methane emissions. And so coastal salt marshes have become increasingly degraded and threatened, with over 30% of Australia's salt marshes being modified to some degree. And some losses have even reached 80% in Southeast Australia that you can see highlighted in the lighter green. And a large proportion of these salt marshes are impacted by agricultural expansion, leading to the construction of tidal restrictions, also known as bund walls. And so bund walls essentially are barriers that restrict any tidal flow. And so this results in decreased salinity on the landward side, as salt marshes often accumulate fresh water through terrestrial runoff and precipitation. So as you can see in the top figure, that would be the left side of the bund wall. And so the resulting land is then converted in favor of ponded pastures for grazing. And so basically the ability of salt marshes to act as carbon sinks and mitigate climate change is offset with major disturbances to their sequestration capacity. And so this could essentially result in their carbon sinks becoming sources of greenhouse gas emissions. So for this study, we aimed to simulate the blocking of tidal flow through bund wall placement and examining the resulting salinity changes on carbon cycling regime. And so we quantified 3 key processes that are involved in carbon soil cycling. And so the first would be greenhouse gas flux, so carbon and methane. The second, organic matter decomposition, so using standardized T litter. And the last being soil properties, such as pH and redox. And so 3 corresponding hypotheses were constructed. The first, that decreased salinity would cause an increased carbon flux and methane flux, since sulfate reduction would no longer outcompete methanogenesis or be a major pathway for microbes, as there is no longer seawater present since there's no tidal exchange. The second hypothesis, that freshwater addition would accelerate organic matter decomposition through metabolic pathway shifts in electron acceptors, which would enhance microbial activity in the sediment. And the last hypothesis, that changes in soil properties would correlate with greenhouse gas emissions and decomposition. So now moving on to the methods. 38 intact soil cores were collected from a salt marsh in Melbourne, Australia. The cores were then left to acclimate in ambient seawater for 7 days in the lab to minimize any effects on greenhouse gas flux. And then was the start of the incubation experiment, which lasted for a total of 30 days. For our experimental setup, so cores were divided into treatment and control groups. And the control group was maintained at 34 ppt to match field site conditions, and the treatment group was diluted to 0 ppt, which would be freshwater salinity. And then the cores were divided into sampling tubs. So there were 4 cores assigned to the greenhouse gas sampling tub, and then we had 15 cores assigned to the tubs for destructive sampling. And so during the experiment, the water level was maintained at 2 centimeters below the soil surface to match site conditions. And now for the freshwater dilution for treatment cores. So to simulate tidal restriction and the subsequent precipitation and runoff, the treatment cores were treated with the addition of fresh water. So we flushed the tub with deionized water gradually once a day from the start of the experiment, every day over 7 days until the end, where salinity was at 0 ppt in the tub. For greenhouse gas flux sampling, so greenhouse gas emission and methane were both measured consistently throughout the experiment with a plastic chamber, as you see in the picture there, placed over the soil core connected to a greenhouse gas analyzer for 7 minutes. And so we sampled more frequently during the start of the experiment to capture any immediate changes to greenhouse gas flux that may have been occurring. And now for destructive sampling, so these occurred on days 3, 5, 9, 15, and 30 of the experiment. And so per sampling, we sampled 3 cores, and they were removed from their PVC pipe and destructively sampled at 4 depths. And so the surface depth, pH and redox measurements were taken. At 5 centimeters depth, pH, redox, and T decomposition measurements were taken. So standardized tea bags, as you see there in the 5 centimeter depth, were used as a proxy for decomposition. And so 1 green and 1 rooibos tea bag were both buried in each of the soil cores at 5 centimeters below the sediment surface before the start of the experiment. And then during sampling, they would be removed, cleaned, and weighed to determine mass remaining. And then we have the 12 and 18 centimeter depths where pH and redox measurements were also taken. For our statistical analysis, so to assess the effects of salinity on carbon flux and methane flux, pH and redox, a linear mixed effects model was used with treatment as a fixed effect and sampling time and replicates as random effects. A single exponential decay rate model was used by fitting proportion dry weight remaining for the green and rooibos tea. And a one-way ANOVA was used to assess the effect of salinity change on mass loss for the last day. Now moving on to the results. So here we have greenhouse gas flux, so carbon. And so this figure here displays average carbon flux emissions across time in hours, with the acclimation period highlighted in yellow. And the dashed reference line represents the average flux at the field site where the cores were collected. And so we have the control group in green, so salty, and then the fresh was the treatment group in blue. And so overall, carbon flux did not vary significantly across treatments. Now moving on to our methane emissions. So methane flux was also not significantly different across treatments. So this result was surprisingly consistent with other studies that have reported increased methane fluxes with decreased salinity. And so the methane fluxes in our study were comparable to other methane fluxes reported in natural salt marshes. Now the green tea for tea decomposition. So after 30 days of incubation, treatment was not a significant effect on proportion mass remaining. And for the decay rate model of the tea, so this figure here displays proportion mass remaining for each tea bag across days incubated. And so the treatment tea bags reflected a higher decay rate at 7.9% mass loss occurring per day, whereas the control had a mass loss rate of 5.4%. So we can see that there was faster decomposition in the treatment cores that had received the freshwater addition. For the rooibos tea, so after 30 days of incubation, there was also not a significant effect on proportion mass remaining, as displayed in the figure on the left. And so for the decay rate model, so tea bags in the treatment cores had a 2.2% mass loss per day, while the ones in the control cores were similar at 2% mass loss. So there was not a huge difference observed for decay rates between tea bags in control versus treatment cores. Now for pH, so this figure here shows average pH measurements by days incubated at the top and depth on the y-axis. And so there was a significant increase of pH in the freshwater cores, as you can observe in the figure, and there was also a pattern of decreasing pH with depth for both treatment and control cores. Observed here range from about 6 to 8, which is consistent with those reported from other coastal marshes. And now for redox, so this figure shows average redox measurements. And there was no significant effect of treatment found, but there was a significant effect of depth, where we can see that redox at the surface was significantly higher than the rest of the depths. So the high redox values we see here at the surface reflect a more oxidized surface layer of the core. And the negative redox values that we see at 5, 12, and 18 centimeter depths are indicative that the soil was under anoxic conditions during the experiment, and the range of values that we see from negative 175 to about negative 300 fall in the range at which methane production would likely occur, but these measurements did not correlate with methane emissions we observed in the study. So now for the discussion, starting off with greenhouse gas flux. So that the experimental design didn't allow for the total flushing of the cores, which resulted in elevated pore water salinities. So we use salinities in the tub as a proxy for soil core salinity. So the salinity might have been decreasing, but not entirely fresh as we wanted. So even if pore water salinities were low, but still at about 15 to 20 ppt, sulfate would still be able to dominate metabolic pathways and outcompete and suppress methanogenesis. For tea decomposition, so the high rate of decomposition we observed in the green tea was expected due to the higher fraction of water-soluble compounds present in the green tea, whereas the rooibos tea has more recalcitrant material that takes longer to decompose. And this is also reported in other studies as well. And the decay in the green tea that we observed could be more related to the flushing of fresh water that took place during the first week of incubation to dilute the salt water. Other studies have observed that water availability and supply can facilitate the breakdown and leaching of organic matter. And for soil properties, so there was reduced emissions from the treatment cores, highlights that there was necessarily direct predictive control of redox potential or pH. The results suggest that salinity changes have minor biogeochemical effects in the short term. However, such effects might become more apparent over the long term. And to conclude, so this study highlighted the initial shifts of freshwater dominated environments under a degradation scenario from bund wall installation. And so we can infer that salinity, microbial activity, and flushing may influence short-term organic matter storage potential. And so the results indicate that maintenance and minimizing disturbance of salt marshes can contribute to carbon offsetting in the context of coastal wetland management and conservation practices. And so a better understanding of salt marsh decline impacts is crucial, as there is a great risk of loss of salt marsh carbon storage functionality. And so this just confirms the need for future manipulative experiments examining long-term impacts. So thank you all for listening. I would like to thank my promoters, supervisor, and the staff at the Blue Carbon Lab at Deakin University for all of their help and support.